So far in class, we've been talking about transcription. And in transcription, we start with DNA and we end with RNA. And the specific type of RNA that we're making is called mRNA. And the M is for messenger. So we're making the RNA that's a messenger of the DNA. And it's a messenger because it can allow the DNA message to leave the nucleus. So remember, we have the DNA hanging out here in the nucleus, and we make a single-stranded copy of the RNA in the nucleus, and then that single-stranded copy of the messenger RNA can go out of the nucleus into what we call the cytoplasm of a cell. Okay, so that's a general idea behind transcription. So when we started, we had our double-stranded DNA, right? And if the DNA read ATGC on the 3' to 5' end, then we're going to make some single-stranded RNA that reads UACG in a 5' to 3' direction. And again, that's single-stranded. So that's just a quick review of transcription. Now we're going to take this single-stranded messenger RNA that's out here in the cytoplasm, and we're going to decode the message. The process of taking that messenger RNA and decoding the message into something called a polypeptide is called translation. Just like you're translating a foreign language. It's like the mRNA is a foreign language that needs to be translated. In this process, we are making a protein, and it's proteins that build you. They build your structures. They give you your brown eyes or your blue eyes, your shortness or your tallness, your left-handedness or your right-handedness. All of those things are determined by proteins. So in this way, the code that is held in the messenger RNA is used to build you and make you who you are. All right, so just a quick review that we're out here in the cytoplasm with this messenger RNA floating around. And some other important things that you need to remember are out here in the cytoplasm as well that we're going to be using. So out here in the cytoplasm, we also have these little structures hanging out that are called ribosomes. And if you remember back when we talked about what different cell structures do, we said ribosomes make protein. And here it is, we're going to be making protein through translation, so we're going to need those ribosomes to do that. Okay, that's where their job comes in. You're also going to see another form of DNA, and this form of DNA is called another form of RNA called tRNA. And this tRNA is also floating around out in the cytoplasm. I'm just going to show it as like little boxes for now. We'll talk more about what tRNA is later. Okay, but this tRNA floating around in the cytoplasm stands for transfer RNA. So this RNA is going to help us transfer something during the process of translation. So out here in the cytoplasm, we have all these mix of different components, the mRNA, the ribosomes, the transfer RNA, and they all work together to make translation happen. All right, in the first step of translation, I'm just going to kind of get you introduced to what these different things are. So this guy right here is those green ribosomes that I was originally is one of those green ribosomes I was originally telling you about. Okay, so that's that. The little guys out here, those are those orange transfer RNAs that I was mentioning. And then finally, this strand with all the letters on it, hopefully looks familiar. 5' to 3', that's our messenger RNA that I was doing in blue. Okay, so now we're zoomed in on the cytoplasm, but we still have these three components. I just want to point out to you for this transfer RNA, we have two parts to every transfer RNA. So each transfer RNA has this kind of top oval part, which is called an amino acid. And then this bottom set of bases, you see right here, okay, so that is where the actual RNA is. They are RNA bases. You'll notice, for example, there's a U here, and there aren't any T's. So a transfer RNA always has two parts, an amino acid and some RNA bases. Okay, that's our transfer RNA. Again, just hanging out in the cytoplasm. So all these different components are together. You get to see them in a little more detail in this first step. Okay, step number 4. Hopefully you can see what's happened here. The ribosome, okay, right here, this big green guy, has come in and locked into place on the messenger RNA. Remember, the messenger RNA is this strand of RNA bases. And it always starts in the same place. This part of our messenger RNA is a very important piece that we're looking for. That AUG is always where the ribosome locks in. We call it a start codon. It's a codon because it's a set of 3 bases. Okay, so any set of 3 RNA bases is called a codon, and it's the start codon because AUG is always where translation starts. That's where the ribosome locks in, finds the right place, settles in, and starts reading the mRNA. Okay, so basically step 1 is the ribosome comes in and locks onto that start codon. Step 2, you can see what we've got here now is a transfer RNA has come over into that place where we had our start codon, and it is matched up with the bases that are opposite of the start codon. So the start codon had A, U, G, and the transfer RNA that came in has U, A, C. So they're opposite. And you'll see sometimes this is written if the mRNA has the codon, which we just talked about. The tRNA bases are called the anticodon because they are the opposite of the codon. And it just so happens that the anticodon, UAC, always codes for the amino acid called methionine. Remember, these are amino acids. There are 20 different amino acids, but methionine is the one that we always start translation with because AUG, the codon, always pairs with UAC, the anticodon, which always has the amino acid met attached to it. So translation always begins with the amino acid met. Okay, in this next image, you can see that another transfer RNA right here has come in. It matched with the codon on the mRNA that has the letters CCA. So again, there's the codon. And it brought in the anticodon, GGU, to be the opposite of the CCA. And the anticodon GGU always brings along with it the amino acid glycine. Okay, so again, that's the amino acid. Now, there is a special chart that I'll be giving you in class that will help you figure out which codon results in which amino acid. But for now, you just take my word for it that these are the amino acids that go with them. One other thing I want to point out at this point is you'll notice that on our mRNA molecule, we've started at the 5' end, and we looked for the AUG coming from that direction, from the 5' end. We're going to be moving down this way from the 5' end to the 3' end. So translation always moves in a 5' to 3' direction. Okay, next image. What have we had happen here? Now remember that the amino acid, the ribosome was originally right here, right? Because we had to find the AUG, okay, and that was the first thing that the ribosome bound to. But now the ribosome has slid down, like I said, in a 5' to 3' direction. It slid on down, and now that glycine transfer RNA is in this furthest left position. It's no longer here. Remember, it was right there before. So since the ribosome slid down, the glycine is moved over. The methionine is still here. Okay, we still have that amino acid, but the anticodon that went with the amino acid methionine has gone away. So we don't have that anymore. But the methionine and the glycine are bonded together, so we're starting to make this chain of amino acids. Next step, hopefully this looks familiar. We have a new amino acid and anticodon combination coming in. Here was our codon, UGC, was the next 3 bases. The anticodon that matches with that is ACG. And then the amino acid that comes with ACG is this threonine amino acid. So we're continuing to add to our chain of amino acids because they're bonding together as they get lined up according to the anticodon. Remember, it's the anticodons down here that carry the message to say which amino acids actually come into play. The next step that's going to happen before I show you the next slide is, again, this whole ribosome is going to slide down. So the threonine that's now right here is going to end up in this position. Okay, so here we are. The ribosome has moved down. The threonine with its anticodon is in the leftmost position on the ribosome. We still have methionine and glycine and threonine bonded together, so we continue to make that chain of amino acids. And you'll see that we no longer have an anticodon here for glycine. That broke off and went away. Those anticodons get used over and over again, so the anticodon for glycine will go find another glycine, bond to it so that it can do this process again. We have an open position here on the ribosome. Our codon is ACU, so the anticodon we're going to be looking for next is UGA, so it's right here. It's going to come into this position on the next screen, and you'll see it fully formed in that one. So there we go. Threonine is still in this left-hand position. Now we've got the correct anticodon in the right-hand position. And you'll notice that instead of having an amino acid up here at the top, it says stop. There are particular anticodon combinations that are attached to a molecule that basically tells translation to stop, which means there are certain codons down here that always code for a stop in translation. So this particular chain of amino acids is done. This is going to be a 4 amino acid chain. In reality, the amino acid chains are much longer than this. They're like hundreds of amino acids, thousands of amino acids long. Okay, but this is just a short one to show you how this amino acid chain is made. So once the process is now done, then we've got our amino acid chain, okay, up here, all still bonded together. You'll notice that there are no more transfer RNA molecules, no more anticodon molecules attached. It's just the chain of amino acids. The ribosome has broken off. Okay, that's over here. Here's our ribosome. So that can be used again to do more translation. We still have some extra transfer RNAs hanging out in the cytoplasm. And we still have the messenger RNA unchanged, so it could be translated again. So that ribosome could hop onto the AUG again and make this chain of amino acids all over again as well. This chain of amino acids we call a poly, meaning many, peptide. And polypeptides are the beginnings of proteins. When we get a really long polypeptide, so I'm just going to pretend there's a whole bunch of amino acids here stuck together. Okay, so there's a really long polypeptide, all those amino acids in a chain, and then we fold it up. So this is my folded up polypeptide. Then we call that a protein. And proteins, as I said before, are what make you who you are. As strangely as it sounds, they are what control you. Not only do they give you your physical appearances, but proteins also, as we've talked about, is what enzymes are made of. It's what those antigens on the surface of your cells are made of. It's what protein channels in your cells are made of. It's what your hair and fingernails are made out of. Proteins are what make you human. And so the instructions to make your protein, which originally came from the DNA and then got passed to the mRNA, are the instructions for making you who you are.